So Arthur, over to you.
Okay. I would like to share my screen. Okay. Well, this kind of actually it started off with Basil calling me one day, and he told me about a particular type of information that he had read, and he was wondering whether I would like to give a presentation on this. So it was kind of interesting. So I went ahead and prepared for it. And the first thing I did, I talked to my son, because he's a physician, and he sort of put all sorts of doubts in me, and it scared the hell out of me. And then after I did everything, I wasn't too sure, so I sent it to him and said, "What do you think?" And he said, "It's very good, Dad. Don't worry about it. It looks good." I'm sorry I put some fear in you. Okay, here goes. Can we reduce cancer mortality by early detection? That's the title. Now, you know, life, the symphony of life, it's all in the DNA, the double helix. Life is like a symphony written in a DNA code consisting of 4 nucleotides, A, T, G, C, with various non-coding regulatory sequences. Similar to the cue from the conductor, it starts with the first cell division and ends in death when all cell division cease. The cell is the building block of the human body. In the cell, inside the nucleus, there are 23 pairs of chromosomes that contain the supercoiled DNA. It contains all the information of the individual. It's all packed in there. It's a blueprint of the person. There are also, you see, lots of different kinds of cells. Cells differentiate. That doesn't mean that they have different sets of genes. No, they all have the same set of genes. What happens is some genes are just, turned off. Other genes are turned on. So if you take a sort of a muscle cell, all the muscle cell genes are turned on. The rest of the genes are turned off. So that's how you differentiate. Now, the first cells are all, they can differentiate in any direction. And several cues are used to make this thing. There are 37.2 trillion cells in the human body. The supercoiled DNA is packed into 46 chromosomes in the form of a double helix. It's 2 meters long and has 3 billion base pairs. There are 20,000 genes and non-coding regulatory sequences that turn on and off the genes. The entire cell has to be faithfully reproduced, including the DNA sequence. And there are 10 quadrillion cell divisions. Imagine, a quadrillion is 10 to the power of 15. Imagine the mistakes in DNA replication that can occur during the myriad of cell divisions. When cells differentiate, only the genes that are required for that cell type are turned on and others are kept silent. The fidelity of DNA replication is of paramount importance. That's how the mistakes occur. When you see the cells differentiate and they multiply, early on they remain the same because they are what are called a pluripotent state where they can become any other kind of cell. And later on, they become specialized cells. The first cell division occurs after the sperm fuses with the egg and forms the zygote. Here you have the 23 pairs of chromosomes. The sperm has 23 chromosomes and the egg has 23 chromosomes. And when they meet, they get the 23 pairs or 46 chromosomes in the diploid state. Now, the cells don't divide at all unless they get a cue. The first cue they get is from the maternal side. And this is called a transcription factor that starts the cell division, turns it on. And this is called ZELDA. ZELDA is the first transcription factor from the maternal side. It turns on the diploid zygote. And then it goes on after that. Sequence of events take place and they divide and divide and divide until life comes to an end. Now, inside the cell, of course, is the nucleus where the chromosomes are packed, and the chromosomes are made of the DNA, and the DNA code has got the A, T, G, C, this, and they are arranged in triplet form, the triplet code for amino acids. Each of these triplet codes, for instance, AAA would be lysine, and CAA would be glutamate, and so on and so forth. The blueprint of a person is written in the form of a DNA sequence. Replication of DNA is therefore of the utmost importance. Every cell should have the exact same copy with no mistakes. Mistakes do occur, and the repair mechanism is present. There is a proofreading procedure to correct mistakes with the DNA polymerase, and those cells beyond repair are destroyed by apoptosis or programmed cell death. The cell itself has a life cycle. It starts off with the first growth phase, and then it goes into the synthetic phase where the DNA is synthesized, and then comes to the second growth phase, and then it divides. It's mitosis, because it's mitosis, equal number of chromosomes are passed on to the daughter cells, and this cycle goes on. Now, during this, there are genes that allow normal cell division. There's positive controls. Genes that allow normal controlled cell division are called proto-oncogenes. These are a whole set of genes called proto-oncogenes. And when they develop mutations, which is actually the mistakes that happen, they become oncogenes and induce uncontrolled cell division, thereby leading to cancerous tumors. There is also a negative control, genes that prevent uncontrolled cell division. They are called tumor suppressor genes, the whole set of them. And when they develop mutations or mistakes occur, they will not be able to stop uncontrolled cell division, thereby leading to cancerous tumors. So these are the two sets of genes that control whether the cell is going to be normal or they're going to become cancerous. Causes of cancer repair and apoptosis or programmed cell death. Proofreading by DNA polymerase corrects small errors in replication. If an incorrect base has been added, the enzyme makes a cut at one particular place called the phosphodiester bond and releases the wrong nucleotide and replaces it with a correct one. In other words, this is a proofreading method. Smaller mistakes are all controlled. Cell proliferation is carefully regulated and mistakes occur, they are corrected. If the mistakes are too far gone and we cannot fix it, they are destroyed by a thing called apoptosis or programmed cell death. Mutations of certain genes, the proto-oncogenes and tumor genes, induce the cells to multiply in an uncontrolled manner, causing a tumor. These cells become immortal. They can multiply and multiply and never die. And if unchecked, they break off and the cells go through the bloodstream, carry to another place, and they will start a tumor in a different tissue. This is called metastasis. So if the tumor is left unchecked, it metastasizes. So you have spreads all over. There are many causes for cancer. Tobacco is a very big causative agent for cancer. Then you have the type of diet you have. Red meat is one example. Sunlight, alcohol, excessive alcohol, poor diet, family history, certain hormones like estrogen, growing older, sexual activity, precocious sexual activity always, very often causes human papillomavirus is transported and they cause cervical cancer. Infections, viruses and bacteria, and environmental exposure of various environmental chemicals cause cancer. Now, programmed cell death is interesting. This is a built-in suicide mechanism that is used the body to get rid of badly mutated cells that are beyond repair. There is a sequence called the death domain, and this domain is activated by a sequence of events which leads to the death of the cell. So this is a normal cell, and when the death domain is activated, there is an enzyme called caspase which starts its activity. The cell shrinks, the chromatin, which is the thing that glues the genes and DNA together, breaks down, and the membrane starts blebbing. In other words, it sort of forms little blebs, and they disintegrate, and everything collapses, and in the apoptotic bodies and the macrophages, the white blood cell, it comes and ingests it and destroys the whole thing. So that's how the cell is destroyed. So in other words, it's so carefully regulated, you know. Small mistakes can be corrected, larger mistakes, the cells are got rid of. But still, these things can escape. DNA, methylation is an important factor. It's the one mechanism that is used by turning off gene expression. So if you don't want a gene to be expressed, it adds this methyl group, and this methyl group turns it off. So that gene is not expressed. And other genes are turned on by transcription factors. But in a cancer cell, abnormal methylation takes place. All types of methylation everywhere. It leaves a characteristic methylation pattern for that cancer. And this is a biomarker. Each cancer leaves a characteristic signature that can be used for diagnosis. DNA methylation pattern for various cancers have been cataloged. So you have a catalog of all these things. You know, this cancer has this pattern, that cancer has this pattern. It's all done there. Now, how do we detect this cancer using this methylated DNA? It's abnormal. Recently, they have come up with a system called liquid biopsy using blood to detect this type of methylated DNA. Now, what happens is when the cell, has been affected by this methylation, apoptosis takes place. And during this apoptosis, the cell is sort of destroyed, but pieces of the DNA escape and they go inside. This is a tumor cell. This is a circulating tumor DNA. These go into the bloodstream because the blood carries all sorts of stuff. Digested food, waste products, various blood cells, all those things traverse. So when a tumor cell breaks down, you get these pieces of what you call circulating tumor DNA. This has that particular pattern, methylated DNA pattern. You can use this, if you can collect this and sequence this and find out what it is, then it will say what cancer it is. Now, but you have only a few pieces of these, not too many. There's very little. And they have the normal cell too, and the normal pieces of DNA. So you have a sort of this camouflage with all sorts of other stuff. Now here is a healthy tissue, pieces come from that. Here is a tumor, and pieces come from there. You have red blood cells, and you have the white blood cells here, and you have all sorts of things. But we want to isolate this particular circulating tumor DNA. That's for detection. Now you can centrifuge it and take the plasma, and that's where you will get these pieces. But isolating this and then diagnosing this, detecting this, is not a very simple process. The analytical validation of this is what you want. Methylated circulating tumor DNA collected from blood samples is compared with circulating tumor DNA profiles collected from cancer patients. So if you compare the two, they can say, okay, this is a colorectal cancer, this is a lung cancer, this is a cervical cancer. Depending on the profile that you compare it with, you can find. The liquid biopsy test can be used for screening methods. And they found out that they can screen more than 50 different cancers, even before discrete symptoms appear. So a person may be perfectly normal, but it's starting the tumor. And if you take the blood and get this particular DNA and compare, you should be able to detect that they have the cancer. It is just starting. Now, in order to do that, you have to collect that and you have to amplify it. Fortunately, now we have very sophisticated methods using immune systems to selectively precipitate that ctDNA, the circulating tumor DNA. And then you can amplify it by the PCR. Everybody knows about PCR now because PCR is used for the COVID tests. And you can amplify it. So you can increase the ctDNA. And then you can compare it to this catalog of various types of cancers and what type of methylated DNA you have. So you collect the cell-free DNA from plasma, and then you sequence it, and then you compare it to the DNA methylation database, then you can use this. Now, with the advent of, various types of machine learning, you can compare this and set this all in a sequence of events, and you can quickly diagnose what they have. The Circulating Cell-Free Genome Atlas study was done on 15,000 participants. When you compare this, you want to find out to make sure that you don't get false positives. If you get false positive, of course, your test is useless. So they try the specificity, the sensitivity, the input titration, repeatability, interfering substance. All these things have to be studied. And when these things they studied, they found out they can detect it close to 99% with certainty. They can say, yes, this is this type of cancer. And interestingly enough, most of the ctDNA or circulating tumor DNA, the early work on the blood sample was done at Princess Margaret. So we can say that in our own backyard, most of the work was done. And I have that reference to the paper that they published in Nature. Grail, a startup company based in San Francisco, collected all this information. I'm sure they must have paid a lot of money to get this information from various sources. And they put this together. I call it a gallery test to detect over 50 different cancers, even before symptoms appear. And they applied to the FDA for use as an early cancer detection screening method. Now, originally, we always have one test, one cancer approach. For instance, for lung cancer, you have a CT of the lungs, and you can detect the tumor. Now, you have a test. One test for many cancer approaches. Now, here you have a list of cancers. The currently cancer screening tests are organ-specific, only 5 types of cancer: breast, lung, colon, cervical, and prostate cancers are detected or screened. Now, with this new approach, the blood-based multi-cancer approach, you can detect more than 50 types of cancer from a single blood draw very early on, even before symptoms appear. Now we'll take another look at these types of treatments that we have. The old treatment is surgery, radiation therapy, chemotherapy. These are well known. But this is more like a sledgehammer approach, where you have all types of non-target tissue is affected. But now we have immunotherapy, which is very target specific. There are hormones that are used, target specific systems, and then stem cells. These are all quite specific. So we have come a long way from the original way of treating cancer. They become very, very specific. Remember, its detection is okay, but cure is possible only if we have a method to cure it. Not all cancers are amenable to cure. So only those cancers that can be treated early on, this system will work. Metastasis is another problem. If you let the cancer progress, pieces of the tumor will get into the bloodstream and lodge itself in different organs. It can, if it is a sort of a cancer from the stomach, pieces can break off, go through the bloodstream, lodge itself in the liver. It can go into the bones, and it can cause secondary tumors. This is what we call metastasis. Now, for instance, here is a 5-year cancer survival rate from 2009 to 2015. Now, if you take the prostate cancer, it's about, you have 30% prostate cancer survival rate in the early on. But if you let it metastasize, 100%, your survival rate, sorry, it's the other way around. The green one is the localized. The brown one is the metastasized. Prostate cancer, if without metastasis, you get 100% cure. But if you let it metastasize, it comes down to 30%. And if you go to this pancreatic cancer, for instance, you can see if it is not metastasized, you get about 40%. It's a very progressive cancer. It's very dangerous cancer, pancreatic cancer. And before metastasis, you get only about less than 5% pancreatic cancer. So metastasis actually is a serious problem. Severely diminishes survival rate. Now, if you take this cancer survival, early detection in general helps in cancer reduction and survival, especially successful treatments are available. New targeted techniques for treating cancer are being developed that appear to be very promising. Liquid biopsies for many different cancers can be detected before symptoms appear and may lead to better cancers. For instance, here in this chart, the dark purple one is a 10-year survival, the medium one is 5-year survival, and the light violet is the 1-year survival. Now let's take pancreatic cancer. If you take this 10-year survival is very, very low. 1-year survival is also pretty bad, 21%. If you take some of the other cancers, say for example, a cervical cancer, you can see the difference. How this early detection is going to play a very important role in reducing the death rate. So this chart really gives you an idea about early detection, what early detection can do. The future of early detection of cancer and treatment. Newer molecular methods for selective circulating tumor DNA extraction, purification, amplification, and sophisticated automated sequence technology have greatly improved early detection method using biomarkers at the nano level. This is the blood test. Learning and problem solving, mimicking cognitive functions, has made the use of artificial intelligence extremely useful. Exhaustive data mining using supercomputers has made machine learning a powerful tool for rapid diagnosis. Now, if you combine these two, and you can have the whole system automated, you will have a very quick way of detecting from just the blood test whether the person has cancer or not, what type of cancer test, and then you can use targeted treatment method. Targeted treatment methods such as use of stem cells and sophisticated immune technology have vastly improved cancer treatments. Tumor formation, metastasis may be common to most cancers, but the pathology varies widely, making some cancers more curable than others. So it's not only early detection, whether it can be cured or not, it's the second one. If you combine the two, early detection serves a very important role. The advent of early screening for multiple cancers combined with newer treatment methods make the future look very promising. Thank you. Now, how do I stop sharing?
Okay. Arthur, that was fantastic. And I have a question.
Okay.
Only one.
Going through this, I have lots of questions. First of all, I'm really happy to know that the funds that we raise for cancer research are actually doing something and making improvements over the years. I see the incidence of cancer have risen, right? Now, has that been because of age or is it because of better diagnosis?
It's both. because as we age now, the survival rate has changed so much, the profile. People live up to 80, 90, 100. So as people grow older and older, the repair mechanisms become weaker and weaker. So when the repair mechanisms grow weaker, the incidence of cancer goes up. So this is age-related. So that is there. We also have the environmental impact, because nowadays we have so many things in the environment. You know, we have various types of plastics that we use. We have, preservatives that we use. These things also have an effect. So when you take all those things, that's there. And the other thing is, of course, diagnosis has become very sophisticated, and we can pick them up at a very early stage. So if you combine all three, you have more incidence of cancer.
Yeah, I see. So if I get a PSA test,
yes,
they always say to you, you may get a false positive.
Yes.
Right. So are they improving that test?
I don't know about the PSA test itself, but the PSA test is supposed to be good. But oftentimes when people always use an escape clause, you don't want to be sort of saying, you say this is possible, but it's also not possible. You put that in just to sort of cover your ass. So that's always there every time when you make a statement, you know.
Yeah. Alan? Yeah.
Hey, Arthur. My question is that you say we can have these blood tests and it will identify 50 different types of cancer.
Yeah.
At what stage would you need to, ask your doctor for this particular test? Or, I mean, there has to be something that would make you say, well, I need a test for these cancer. Let's see if I've got any cancer in me. Because on the normal blood work that I would get on a regular once a year or so, they wouldn't do these sort of tests, right?
No. This is not even available right now. The FDA approval has not come through. But the thing is, if you're above 50, then chances are, and if this is available, they project it may cost about $200 or so to do one test. And if the healthcare people think this is useful to cover this screening, then it may become a sort of a useful tool. So early screening, because they can treat it well, and especially if you have some immunological method for treating it, then you can be cured. So that can happen. So I don't know, about 50-ish when they can do the early screening. It's an arbitrary number that I'm choosing.
On the normal circumstances, they would not, if you went to your doctor, unless you weren't feeling well, he's not going to send you for this sort of test then.
No, normally you won't if you're a healthy individual. But, there are certain things that can be, if you have a family history of cancer thing, then this might be a useful technique. For example, people who have a certain type of gene, BRCA1 and BRCA2, these are breast cancer genes. If these two genes are present in a person, chances are that person that can get breast cancer. Now, the case of Angelina Jolie. She has both the genes, so she had a double mastectomy. It's a sort of a prophylactic way of controlling breast cancer. So she lost both her breasts. So now what you see is a prosthetic breast. That's what you see.
Dave?
I have to jump into another meeting at 1:30, but thank you, Arthur. This is great. As you know, on our next normal podcast we're looking at the future. And Arthur, I was really struck by the computing requirements to find out what the results are. We've been looking at Tencent, the Chinese company, has recently developed a quantum computer that, they had IBM's Big Blue. They challenged it to a problem that would take 2.5 billion years to solve. And Tencent's quantum computer solved it in a couple hours. So just in terms of where we're going, in terms of diagnostic and the computing power for machine learning, it's quite positive actually. So
this machine learning is going to revolutionize everything, because what instead of doing one test after another test after another test, if they have everything processed by artificial intelligence, where it's all stored and the machine can sort of, do certain things and make decisions that a human is not involved. And the machine will have no bias. It'll automatically search its stuff that it has stored in its memory and make conclusions. And it'll say, okay, this is what it is, and diagnose the particular type of cancer. So they have this DNA methylation profiles for all these cancers. They are cataloged and is available, and they feed it to this machine. And, and the artificial intelligence system will use it for diagnosis, which is just fascinating.
Yeah. Thanks, Arthur. I got to go. Take care, folks.
From a cost containment sort of idea, it's worth spending that $200 because the cost of treating a cancer is like 1,000 times more expensive, right?
Exactly. So if they have a routine thing, and if they have it all built in, as they make use of it more and more, the cost will come down. And then you can see, if you project it and see how much it will cost to treat the cancer, if you catch it early enough, you're better off.
Basil? And maybe one more after that, in the interest of time.
Arthur, when I read about this blood test for cancer, I knew that there was only one person in our club who would be qualified to do the research necessary to talk to us intelligently about it. You've done an amazing job. I know you were a little hesitant, and you question whether it's something you would have time to undertake, but as usual, when asked, you always come through, and this was fantastic.
Basil, you won't believe me.
Last one is Courtney.
I read more than 200 papers, which I got from the University of Toronto Library, so that I can sort of be sure of what I'm saying. I certainly didn't want to make an ass of myself.
Well done.
Okay, Courtney, your question, and then we'll get Trudy to thank Arthur.
I'll keep it quick. Arthur, at the risk of asking you to repeat yourself, and a timeline for when this kind of blood test could be available here in Toronto, across Canada?
Right now, the FDA is reviewing it. So, and all the indications are that they'll grant the permission to use this, and they'll be licensed. So if the FDA, approves it, because the Americans always plunge into it right away, we are a little more careful in Canada. Health Canada will look at it. Don't forget, Princess Margaret is here with all the background information. So our reviewers are well placed to look at it very critically. And if it is done in the US, probably another 6 months after that. I would say in a year, we should definitely have some information on it.
Right. Okay. Thank you. Excellent.
So Trudy, if you could thank Arthur, please.
Trudy.
You're on mute. Yeah, that's it.
Arthur, thank you so much. I know that I don't love science myself, right? Because I feel like it's so complicated, right? But every time you have presented something, you kind of put it in the layman's language and break it down very easy to understand. So thank you for the time and the research and all the papers that you have read to really bring to us such an informative and well thought and researched presentation. Thank you so much, Arthur.
Thank you.